Cell heterogeneity is always present to some extent within any cell population, be it cell type or function, where a cell is in its cell cycle, as well as differences at the biochemical or genetic level. Seemingly identical cells can differ greatly in their response to changes in environment, and these differences can have important consequences for both health and function of the entire population. Importantly, we should be aware that disease states such as cancer are highly heterogeneous, which goes some way to explaining why there is such a diverse range of pharmacological responses to therapeutics in cancer treatment. Being able to analyse at a cell-by-cell level allows for the study and identification of subpopulations that could potentially be overlooked within population average data analysis. So what are the current techniques used for the analysis of cellular subsets, and what kind of biological insight and productivity do they provide? Flow cytometry has the ability to take large numbers of cells and, coupled with the use of fluorescence, separate cells by gating into particular subsets depending on their marker expression as well as size and complexity. And this technique is becoming ever more high throughput with the help of platforms such as the IncuScreener. However, because flow requires the lifting of cells in order to put them through the machine, it doesn't take into account the morphology of the cells. In order to do this, you need microscopy. High-content imaging uses automated microscopy, and image processing allows for high-throughput screening, which can again be used for subset identification depending on morphology and fluorescence. However, this uses endpoint fix and stain methods, so it doesn't afford the user any sense of kinetic data. Time-lapse microscopy is able to provide kinetic data and can be used to monitor cells label-free using a specially designed microscope stage. However, this is mainly single cells being tracked over time, and so cell sampling sizes are small and throughput is low. These are all powerful methods for studying the subject of heterogeneity. However, there remains an unmet need for a technology which provides high throughput, in-depth live cell imaging that is truly kinetic. IncuCyte provides a novel integrated solution for real-time automated image collection and the analysis of cell biology. It is a well-established method for quantifying phenotypic changes in cells. It's able to do this as the IncuCyte sits in a standard cell incubator, so the user is able to fully control the cell's environment whilst automatically capturing and analysing time-lapse images. There are a wide range of assays for quantifying phenotypic changes, including cell proliferation, apoptosis, and immune cell killing, to name a few. So far, IncuCyte analysis of these assays has been based on population average data, whereby information from all objects or cells in the field of view is consolidated into an aggregate metric. While this type of analysis is a powerful tool, and such measurements can accurately capture a population state and any variation in it, it fails to differentiate a large effect on small subsets in the greater population. Changes in these small subsets are masked by the greater number of cells in the so-called non-reactive populations. The main content of this webinar is to introduce our new software, which is capable of analysing at the cell-by-cell level, and we believe this development is an important addition to the analysis technologies previously described. So I'm going to first explain how you would set up an assay, and then how you would go on to analyse it using the cell-by-cell analysis. The preparation of the assay plates is performed as usual, where you would seed your cells, add treatments, and include any non-perturbing agents, then place in the IncuCyte for image acquisition. And this acquisition step is all done using the 20 times objective. Once you have acquired your images, you can then set up a cell-by-cell analysis processing definition. Currently, this software is optimized for non-adherent cells only. The analysis allows for the segmentation of the phase image and subsequently identifies the cell boundaries. And with this first part of the analysis, you acquire your total cell number metrics. Now you have identified your objects, you can then measure and classify them based on metrics such as size, shape, or fluorescence properties. You can see from step 4 that the gating tool uses a similar concept of flow with the 2D dot plot. Once you have applied your gates, you can then visualize your data in the form of time courses, where you can observe how your chosen subset changes over time. So this video nicely puts together the process of the last couple of stages that have just been described. The assay setup here uses red-labelled cells in the presence of a cytotoxic agent, and our caspase-3 reagent has been added which lights dead cells up green. So here we have the 2D plot where our cells, or events, are represented by these dots on the graph, and we have red fluorescence signal on the x-axis and green on the y. A quadrant gate has been applied to the plot where the bottom right square shows events with a positive red signal representing all of the living cells, and the upper left showing cells positive for a green signal when they're dead. Over time, you can see the cell population moving through the quadrants, starting where the majority of cells are just positive for a red signal, but then as the cytotoxic agent starts to kill the cells, the population moves into the upper right quadrant, where cells are in the process of dying and so are both red and green, and then finally into green when they are no longer alive and are not able to produce the red fluorescent protein. From this population gating, the information can then be used to produce time courses of your data's behaviour, with the added advantage of being able to see your gating applied to the images, which you can see here on the far right. This gating or backgating feature allows you to colour-code your subsets and overlay this gating back onto the original images, so you can also observe how your subpopulations are interacting with each other. Now I'm going to go through some validation data for the cell-by-cell analysis, as well as some examples of how you could use this capability. We validated the phase segmentation by comparing cell count acquired by cell-by-cell analysis to nuclear count. The cells here used in these images on the left-hand side are Jurkat nut-like red cells, and they have been plated at various densities and then monitored and analysed over a 4-day period. Each image was analysed using cell-by-cell analysis, which is here in the top right image, with the mask applied in yellow, and the same image was analysed using nuclear counting, which is shown in the bottom right-hand image, where the mask is applied in green. In the far right panel, the data from each analysis method is shown on the graph with phase count, represented by closed circles, and nuclear count, or NUC red, by open circles. As you can see, each dataset is highly comparable and overlays the other almost exactly for all of the cell densities and for the entirety of the experiment. This result was also achieved using a number of different non-adherent cell lines. The ability to use a label-free method of cell counting really does benefit the user greatly and allows for truly uncompromised live cell assays. This next slide is validation for the use of cell-by-cell analysis itself and outlines the advantages it provides the user. So, still using solely the phase masking abilities that have been afforded by the new phase segmentation algorithm, we can already extract a great amount of data, which includes information on a cell's morphology. The image on the left shows activated PBMCs that have been individually segmented by the new software, and this enables the user to measure size using area and shape using eccentricity. Eccentricity refers to roundness of a cell, where an increase in the arbitrary unit means the cell is becoming more elongated. The two graphs here show how the data looks when these values are presented as an average. The control population is non-activated PBMCs, which are shown in grey, and activated in blue. The graph on the left describes an increase in the eccentricity of activated cells, and the graph on the right shows there is also an increase in size, both of which are hallmarks of activation. Looking at just these two graphs, you could assume that by the final time point, the activated cells were a homogeneous population that have all become large and elongated. However, the true power of using cell-by-cell analysis is being able to look at the distribution of the data. The images along the top show the difference between the non-activated and activated population over the course of the experiment, and we can see in the image on the right there is a clear increase in heterogeneity within the population in regards to cell size and shape. When you plot all of these cells as individual events below on the 2D plots, these differences become even more apparent. At the start of the experiment, we can see a very homogeneous population. Post-activation, we do see the large and elongated cell subset as described in the previous slide. However, it is very obvious that there are a great deal of other cells that do not fall into this category, and we can see, using cell-by-cell analysis, the total population is actually highly heterogeneous. Only with cell-by-cell analysis is the user able to view this data in deeper biological detail. Now, aside from differences in morphology, PBMCs themselves are made up of a host of various different immune cells, and using phase alone, they cannot be distinguished from each other. For further subset identification, fluorescence must be used. We have a method for fluorescently labeling cell surface CD markers on live cells using our FabFlour-488 antibody labeling kit. The antibody raised against the particular marker of interest is firstly mixed with our FabFlour-488 reagent, which fluorescently labels said antibody. The Fab is raised against the Fc portion of the antibody, so the labeled antibody is still able to function and bind to the marker. The labeled antibody is then added to the assay plate together with a fluorescence background suppressor and any other reagents necessary for the assay. Lastly, the cells are added to the plate, and then the assay plate is placed in the IncuCyte for imaging. The antibody is then able to bind to the marker as and when it appears on the cell, allowing for the identification of further subsets. And this is a one-step, no-wash or separation protocol, and it's non-perturbing and compatible with IncuCyte optics. This is an example of some cell staining using the protocol just described. The left panel shows the phase image of Ramos cells, and in the middle image we see them fluorescently labelled with the CD20 antibody together with the FabFlour reagent. The right image shows the phase cell-by-cell mask overlay, and the fluorescence properties inside this mask are added as a metric that can be used for gating. This graph shows the number of Ramos cells positive for different markers as a percentage of the total population, including CD45 and a control IgG to exemplify that this labelling is both specific and long-lasting. This next slide further validates the ability to detect subsets using fluorescent markers. Here we use mixed populations of Jurkat and Ramos cells and labelled with a CD20 antibody. CD20 is a B cell marker and is only expressed on the Ramos cells. The images on the far left show a 50-50 mix of the two cell lines labelled with CD20. You can see the above image shows roughly 50% of the cells are labelled, and the image below has the backgating applied, where the pink outline represents cells that are positive for CD20, and the blue lines highlight cells that are negative. The middle panel expresses the CD20 positive population as a percentage of the total population, which corresponds to the original percentages of Ramos cells added to the mixes. CD45, which is present on both cell types, was therefore represented by 100% of the population regardless of cell type or proportion. The panel on the right shows how the CD20 positive population can be tracked over time, and the proportions also remain the same. Just to note that there is an increase seen in the actual percentage over time in each of the mixed cultures, and this is purely because the Ramos cell line has a more rapid rate of proliferation. Conversely, the negative population could also be identified and graphed in order to describe the Jurkat population. We can also take this example further using two colours. This is again using mixed populations of immortalised cell lines, these being Jurkats, which are a T-cell cancer cell line, and THP-1 cells, which are a leukemic monocyte cell line. Here, in the upper left image, we have identified the CD3 expressing cells, which are the Jurkats, and these are green and outlined in blue, and the negative population outlined in yellow. An X in 5 red has also been added, which identifies dead or dying cells, which you can see in the image to the right. To these mixed populations, we added Fas-ligand, which specifically kills T lymphocytes. The bottom two panels show how the dot plots are used to separate the dying Jurkat subpopulation, which are CD3 positive and a Nexin positive. In the graph on the right, the numbers of dying Jurkats expressed as a percentage of the total population are proportional to the known percentages that were added to the THP-1 cells at the beginning of the assay. It is also important to show that this method of measuring protein expression is comparable to technology that already exists, and so IncuCyte data was compared head-to-head with that of Flow Cytometry. PBMCs were isolated from a number of different donors, as shown in the table on the right, and immunophenotyped using either the IncuCyte FabFlour reagent coupled with IncuCyte imaging and analysis, or a flow cytometer using the standard antibodies and protocols. It can be clearly seen from the graph on the left, the numbers between these two techniques marry up nicely and show a strong correlation between the two methods. Now we can return to what was previously discussed in regards to the heterogeneity of activated PBMCs and focus on further analysis using both morphology and fluorescence. FabFlour-488 labelled CD71 has been added to this activation. Upregulation of CD71 expression on CD8 positive T cells is an early marker for their activation. Cell area and green fluorescence have been used to construct 2D plots as shown on the left. At 0 hours, the majority of the population is again very homogeneous, where cells are small and do not express CD71. After 120 hours, we see the population becomes very heterogeneous in both size and marker expression. The middle graph shows the overall expression of CD71 is increasing as a percentage of the total population, and the activation appears to cause upregulation to CD71 specifically. If we use CD4 as another example, this marker increase is significantly smaller. The graph on the far right then shows this expression in more detail and shows that it is the larger cells that are preferentially expressing CD71 rather than the smaller PBMCs. So by using the power of cell-by-cell analysis, we can identify individual subpopulations that are responsible for the increases in various marker expression and track this subset over time. We can also use marker expression to show how subpopulations respond to biological stimuli in different ways, and also track these changes in subsets over time, and importantly, link this to function. The images on the left show PBMCs activated with interleukin-2 and anti-CD3 antibody. The top image shows CD8 positive T cells, and the image underneath CD4 positive T cells, both highlighted in pink by the backgating tool. The graph on the left shows the cell count in response to increasing concentrations of anti-CD3 antibody. Proliferation in response to activation with anti-CD3 antibody shows total cell numbers increase in a concentration-dependent manner. The histogram on the right, showing green fluorescence intensity at 120 hours, gives an indication, however, that each cell type is behaving differently, as it is clear that the positive signal for CD8 is greater in comparison to the CD4 signal. If we then look in more detail at the data using cell-by-cell analysis, we can then show the percentage increases in both CD8 in the left panel and CD4 in the middle panel. The graph on the left describes how CD8 positive cells are much more responsive to treatment with CD3 antibody. At the beginning of the experiment, both CD8 and CD4 positive cells account for approximately 20% of the total population. There then remains a lag period of approximately 3 days, and post this we see an enrichment of the CD8 cells to over 60%. The graph in the middle shows CD4 cells do respond to the CD3 stimulation, and there is a similar temporal pattern. However, the result is much less, with only 40% enrichment. We can extrapolate this data to suggest that increase in total cell proliferation, as seen in the previous slide, is mainly due to the CD8 cells. As well as inducing CD8 proliferation, stimulation with CD3 also causes the cells to become activated, where they will seek out and destroy antigens. Using the same stimulation conditions in an immune cell killing assay, the graph on the right shows that target cell death aligns temporally with the CD8 proliferation data, which suggests the enrichment of the CD8 subpopulation serves to attack and kill any target cells present. This film on the far right shows CD8 labelled cells interacting with cytolite labelled SCOV3 cells, which are a cancer cell line. Once the CD8 cells have detected the target cell, the T cell then creates an immunological synapse between the two cells using the pore-forming protein perforin. This then allows it to pump apoptosis-inducing proteins into the target cell in order to induce death. This example shows how IncuCyte technology can be used to identify and monitor change in a specific subpopulation and then link this to a functional output. So, in conclusion, IncuCyte cell-by-cell analysis provides an integrated solution for identifying and quantifying subsets of living cells in heterogeneous cultures over time. The module enables temporal analysis of cell subsets based on morphology, biomarkers, health and function. The assay setup does not require cell lifting, fixing or staining, and the simple experimental analysis workflows are amenable to the everyday user. This module provides a further dimension to live cell analysis for non-adherent cells where the user may answer biological questions at the cell-by-cell level.